This is the second part of the chapter 7 note. This is on cell transport. Back for a second. The structure of the cell membrane is essential to its function of being selectively permeable. Remember, selectively permeable means that certain things are allowed through the membrane and certain things are not. The membrane itself selects for what it allows across the membrane. Nonpolar molecules, remember, polar means that one end of the molecule is slightly positive in charge and one end is slightly negative. An example of this is water, where the oxygen atom is slightly negative in charge and the hydrogen atoms are slightly positive in charge. That's what polar means. So nonpolar are things that do not have this. Are hydrophobic, meaning they don't like water, and can therefore dissolve in the lipid bilayer of the membrane and easily cross it without the aid of proteins. Polar molecules, so things like this, and ions, those are going to be things that have a charge. Remember from chapter 2, those are some examples. Are hydrophilic, and the cell membrane does not easily allow these things to pass through the lipid bilayer. Hydrophilic substances can bypass the cell membrane's lipid bilayer by passing through a transport protein. We discussed those in the first part of this chapter. They are embedded in the membrane. Channel proteins are one class, and they simply provide a tunnel through the membrane. Channel proteins do not require energy. They're just a tunnel. They're not a pump. Aquaporins are an example of this, and they allow for water to pass through, because remember, polar molecules need help, and water is a polar molecule. Carrier proteins hold on to molecules and change shape in a way that shuttles them across the membrane. These are specific to certain molecules. So they change shape, that's important, and they shuttle them across the membrane that way. The channels are just a tunnel. They do not change shape. They just provide that tunnel. Diffusion is the movement of molecules of any substance so that they spread out evenly into the available space. So this is not necessarily talking about the cell membrane. Diffusion can occur in a beaker of water. Make a beaker of water here, and then you put some drops of red dye in it. They will move throughout that water until eventually it becomes uniform. That's what we call equilibrium. Movement is random, but as a whole, movement is directional. So this is kind of a hard thing to think about. When particles move, they move randomly. It doesn't really matter how they're moving or where they're going. However, if you put this dye in this water right here, it's going to have a directional movement in a certain direction because you have to diffuse it till it reaches that equilibrium. We'll show a picture here that might make a little more sense soon. Movement will be from an area of high concentration to an area of low concentration. And I designate this as plus and minus. Down the concentration gradient. Dissolved oxygen diffuses across the cell membrane. That's an example. It does not require the cell's input of energy. Okay, so it's again a spontaneous process. It would have a negative delta G if it were a chemical equation. Let's see. Dissolved oxygen is something that can easily diffuse across the membrane. Why? Because it's small, it's nonpolar, and it's not an ion. These are the things that can typically diffuse across the cell membrane. Things that are small, nonpolar, and not ions. Now, what do we mean by small? Well, oxygen only has 2 atoms. That's pretty small. Glucose can diffuse across the membrane. It's pretty small. It only has 12 plus 12, 24 atoms total. Now, proteins cannot diffuse because proteins are made out of amino acids, and each amino acid is probably going to have upwards of 20 amino acids each, or 20 atoms each. Multiply that by hundreds of amino acids to make a full protein, and that's going to be much larger than glucose or oxygen. So this is what I was talking about, the molecules of dye. These individual molecules, this movement is random, but overall they're going to have a net movement towards the side that has less dye, from high concentration to low, until we reach equilibrium where both sides are roughly equal. The molecules are not going to stop moving. They're still going to go across the membrane. It's just that for about every one that goes this direction, one will go this direction. Then another one will go this way, another one will go that way. So no net movement is what we say once we've reached equilibrium. And that's why we're using the word net here. What's the net diffusion? Because even though all of these particles here are going this way, that doesn't mean that some of them aren't going back that way. Okay? The net is the overall. Osmosis is the diffusion of water across a selectively permeable membrane. To discuss water movement, we need to think in terms of tonicity, the ability of the surrounding solution to cause a cell to gain or lose water. That's what this is. Okay, and you've heard these terms probably in Biology 1. It may have been hard to remember them. You're going to have to remember them this year for sure. So let's kind of talk about what they mean. Isotonic is probably the easiest, no net movement. So this is when roughly we are at equilibrium. We don't have any net movement. Hypertonic solutions, there's more solute. How do I remember this? Remember this because hyper means more. For example, if you are hyperactive, you have more energy. So it has more solute. This is the key here. This is where people get confused. More solute means what's not water. Now, that might be salt, might be sugar, it might be a protein, it could be any number of things. It's whatever's not water. So more solute. In a hypertonic solution, a cell will lose water. So if you put a cell in a beaker and the solution is hypertonic, the cell is going to lose water to that solution. There's another way I've heard this saying is that, think about it like this. More solute would be out here if it's hypertonic. Salt is a solute. So if you put a cell in a highly salty concentration, salt sucks. Salt is going to suck the water out of that cell. Hypotonic is when you have less solute. People that are hypoglycemic have low blood sugar. Less solute concentration. In a hypotonic solution, a cell will gain water. So put that same cell into a hypotonic solution, and the water is going to rush into the cell. This shows something. This is called a U-tube, not the online website. A U-tube is a apparatus that has a selectively permeable membrane separating the two sides. And what's nice about a U-tube is it allows us to look at how molecules move by measuring the volume of water. So here we have a bunch of sugar molecules. In relation to the other side, there are more sugar molecules on the right side of this U-tube. Okay, so that means there's more solute. There's more solute, the concentration is hyper or hypotonic. hyper. Good. Okay, so water is always going to move. Now, this is a semi-permeable membrane. We're assuming here that the sugar molecules are not able to get through there. They're too large. So, the only thing moving here is going to be water. Water always moves hypo to hypertonic. Water is going to move from high water concentration to low water concentration. The water is going to move this way across that cell membrane. Well, because we have this U-tube, it's going to be very easy to see that because the water level is going to rise on this side and it's going to fall over here, showing us which direction that water is moving. And this just shows a close-up here of that membrane and what's actually happening. You'll see here that the sugar molecules are actually too large to fit through there. But the water molecules are not, they can pass through. So this is showing an animal cell and a plant cell. A plant cell in the same type of solutions. And this is interesting because animal cells and plant cells behave differently in different solutions. In an isotonic solution, if you put an animal cell in that, water is going to move evenly in and out. That's normal for an animal cell. You place an animal cell in a hypertonic solution, so maybe something that's really salty. Remember, salt sucks. It's going to suck water out of this cell. That's going to suck water out, and that's going to cause the cell to shrivel up. Okay? Now, in an animal cell, that's bad, but in a plant cell, that will actually cause the cell membrane to what we call plasmolize. That means it pulls away from the cell wall. This is that rigid outer cell wall here. This is really bad. This is when you would see a wilting plant. We go over the hypotonic solution. You put an animal cell in a hypotonic solution, all this water is going to rush into that animal cell, and it's going to swell. It could actually burst. We call that lysing. That's not good. Now, for a plant cell, that's actually normal. That's what causes the plant to stand upright. It's because it has a lot of water in all of those plant cells that keep the cell wall nice and rigid, the cell membrane out here. Now, why can a plant cell be normal here and an animal cell actually burst? Well, part of it's that cell wall. And secondly, plant cells have this vacuole. Remember? And that vacuole holds and stores a lot of water for the plant. The animal cell doesn't have that, and so it's going to be more prone to bursting, that and the fact that it doesn't have a cell wall. Hypertonic and hypotonic solutions can be problems for animal cells that do not have that rigid cell wall. Many organisms that live in water are equipped with mechanisms to regulate water. Example, they have cell membranes that are less permeable, so they have less movement of water in and out. Some of them even have contractile vacuoles that pump water out of the cell so that it doesn't get overly waterlogged, I guess you could say. We'll talk about this in Unit 6, but we have things that are called osmoconformers and osmoregulators. Osmoconformers are fish or things that live in water that typically have the concentration in their cells that is the same as the solution. For example, a jellyfish lives in salt water, might have a saltwater concentration very similar to its surroundings. It conforms to those surroundings, so that osmoregulation is not, or so that osmosis is not a big problem. Osmoregulators actually have things like contractile vacuoles or other means to regulate the water concentration in their cells to account for the difference between their body cells and whatever the surrounding solution is. Plants, fungi, and some prokaryotes, remember these are things like bacteria, and protists, unicellular eukaryotes, have cell walls which helps to regulate the water balance. The cell wall will provide a back pressure when it becomes too full. This is called turgor pressure, and the cell becomes turgid. So that's normal for plants to be turgid, to be very, very full of water. Let me show you a picture back here a second. So this is your normal turgid cell. So the water obviously puts pressure on the cell wall. But the cell wall is also, the inside of the cell is putting a pressure on the inside. So that's what we call like back pressure. So there's pressure here and there's pressure here. And that allows it to maintain that turgid stability. Now we'll talk more about this back pressure when we get to calculating water potential. Because one of the things we'll have to talk about is pressure potential. Most plant cells are normal in the turgid state. This is the hypotonic solution. Remember, normal for an animal cell is going to be isotonic, but for a plant cell, it's going to be hypotonic. In an isotonic, in an isotonic solution, plant cells become flaccid or limp. That's because they undergo plasmolysis, which I talked about, when the cell membrane separates from the cell wall in a plant cell because the vacuole is not holding enough water. It occurs in a hypertonic environment. And that's that picture again. Facilitated diffusion is passive transport through a channel. So, for example, water goes to the aquaporins. So water is osmosis, but it is technically a facilitated diffusion. Or it can go through a protein carrier in the cell membrane. Scientists are still trying to understand how these work without energy. Most are specific to what they transport. For example, the aquaporins transport water. Ion channels transport ions. Some are gated channels which open and close in response to stimulus. That can be electric or chemical. Most ion channels are going to be allowing things to go high to low. And some ions, or most ions actually, are going to be more of an active transport, but there are some that go through a facilitated diffusion. Remember that passive transport means that there's no energy required for the process to happen. And remember that cellular energy is ATP. This is just interesting. Cystinuria is the absence of a protein carrier that transports cytosine and some other amino acids across the membrane of kidney cells. The result is painful kidney stones from the amino acids and accumulated from the amino acids that accumulate and crystallize in the kidney. That's kind of interesting. Cystic fibrosis is another one where there's a, I can't remember the name of it, but it's a specific protein channel that is not operating. It doesn't transport chloride ions, I believe it is, efficiently, and so that causes the mucus buildup that is very characteristic of cystic fibrosis when they have mucus buildup in their lungs and they can't breathe. They have respiratory problems. This is just showing a protein channel where facilitated diffusion is just allowing a channel there because these molecules, for whatever reason, can't go through that membrane. Maybe they're polar or something. And this is how a carrier protein works. It's a little different. Remember, the protein channel is just literally a channel. And then a carrier protein actually changes shape. So the molecule specifically fits there, and then it changes shape and releases it on the outside. Whereas the channel is just a channel. It doesn't change shape at all or alter itself in any way. Active transport means the pump against the gradient, and it must expend energy. So it has to use ATP. They use carrier proteins to get things across the membrane. The most famous active transport example is a sodium-potassium pump. An animal cell has a much higher concentration of potassium and a much lower concentration of sodium. The membrane helps to maintain this steep gradient by pumping potassium into the cell and sodium out of the cell. Well, why would we want to maintain that gradient? It actually has to do with your neurons and neurons firing and cell signaling, which we will learn about in around Unit 6, I believe. We'll talk about that. But there's a reason that it keeps this gradient, and it has to do with an action potential in a neuron. ATP is the energy source for active transport. Here's how that sodium-potassium pump works. So this is the cell, and it's going to attach the sodium ions in the pump. Once 3 of them are attached, remember, see, this is that where the ATP comes in because it needs the energy. The carrier protein is going to change shape and expel these 3 sodium ions on the outside of the cell, on the what we call the extracellular matrix. Then over here, the cell is going to pick up 2 potassium ions, and then it's going to change shape, and it's going to release those potassium ions into the cell. Potassium ions. This just shows kind of a summary of the types of transport that we have talked about. Diffusion, right through the membrane. Facilitated diffusion either through a protein channel or a protein carrier protein. Both of these, no energy required. Active transport, energy is required, and it's typically going to move things from low to high concentration, and it's going to expend energy to do that. This, no energy, and we're usually going high to low concentration. A membrane potential is where ions charge has a difference across a plasma membrane, and that creates a voltage difference. Kind of think like a battery, which is the membrane potential. This favors the diffusion of ions across the membrane. This is important again in a neuron. So notice that sodium and potassium both have a positive charge. The reason, even though they both have a positive charge, this is the outside of a cell, this is the inside of the cell in a neuron. They both have positive charges, sodium and potassium. However, there is a more positive charge on the inside of the cell and a less on the outside. And then what happens is as the action potential is going along the neuron, it will actually switch this charge so that the inside becomes more negative and the outside becomes more positive. And then it will go back and remake it so that it's like its original. So when we say it has a voltage difference, it may not even be that there's a lot of things that are negative out here. It might just be that there's less positive than there is here. Think about it like, I'm trying to think of how to explain that to make it make sense. I don't know. I'll come up with something later, but just think of like metal. You have all this metal in a jar, and you have metal out here. You got pennies out here, and pennies in here, and it's not that there's no metal out here. It's just that maybe you have more metal in here, so this is has a higher concentration of pennies. Maybe. Not that there's none out here, but this just has more. It's kind of the same way here, and I'm not sure that this is really the best example in the world. I wish I had a better one, but off the top of my head, I can't come up with anything. When we're looking at a voltage difference, It may not be that one is really negative and one is really positive. It may just be that one has more positive than the other. And for some reason that's not taking away my writing. Let's see if I can get it to go away here. Okay. Co-transport occurs when a co-transfer couples the downhill diffusion of one substance and uphill transport of another. The example here is a sucrose proton transporter in plant cells. It has aided medicine in creating treatments for diarrhea. Given a solution with high salt and glucose, solutions are taken up by sodium glucose transporters so the individual can absorb the water. So, Gatorade. Power drinks. Given this solution, the high salt and the glucose, and the solutions are taken up by the sodium glucose transporter, I'll show you here in a minute, so the individual will be able to absorb more water. This is why you might hear people tell you if you have diarrhea to go drink some Gatorade. And that's actually true because pure water isn't going to do this. It's not going to help the sodium glucose co-transporter. And if you're really dehydrated and you go to a hospital here in America, they're going to give you an IV solution. And they're going to give you an IV solution that is not pure water. It is going to have some salt and some sugars in it. And the reason that they do that is because of this phenomenon. They know this. And when you think about it, think about osmosis. If you pump pure water into your bloodstream, all that water is going to be very, this is your blood, and these are your blood cells, or the rest of your cells, or any cells in your body, brain cells even. If you have, you put a bunch of water, it's very hypotonic, it's all going to rush into those cells, and it's going to cause them to burst or lyse. That's why people that drink pure water when they're running, and they drink lots of pure water, gallons of it, they can actually have seizures and headaches and things because they're causing those cells to burst. What they actually give marathon runners is something that's similar to glucose or a Gatorade solution or a pack that they can put in their water that has some salt and some glucose to actually activate this. In foreign countries, they do give them a drink that is like this. The reason that we put it into your IV in America is because we can afford to do that, and it's actually faster. But they don't ever put pure water in your veins. They will probably say like a lactate bringer is an electrolyte solution, something like that. So here is the example of the sucrose proton co-transporter. So energy is required here for this proton pump. This is going to pump lots of protons up here. That's going to cause that voltage difference. So even though over here doesn't necessarily have a bunch of negatives, it has less positive ions than the side over here. This voltage difference here, this extra H plus, is going to cause sucrose to be absorbed here, okay, into the cell. This is the inside of the cell. It's going to cause it to diffuse this section. That's what a co-transporter is. You need one to help the other. Okay, the last topic is bulk transport. Large materials are packaged into vesicles, which fuse with the cell membrane. It's going to require energy because it's still active transport. And there are two types, endocytosis and exocytosis. Endocytosis is anything that's going into the cell, and exocytosis is anything that's going out of the cell. Technically, endocytosis has several different types. Phagocytosis is large particles that are things like food, things that are particulate. Pinocytosis is anything liquid. And receptor-mediated is actually where receptors on the cell mediate that process. I'll have you watch a video here in a second. This is a picture of that. So phagocytosis taking in particulate matter, pinocytosis taking in liquid matter, receptor mediated. Here's receptors, and these molecules must attach to the receptors in order to initiate this vesicle forming on the cell membrane. It's pinched off, and then the particles are inside that vesicle in the cell. Okay. Okay, in your notes, I want you to first watch the animation on the sodium potassium pump and write down some notes from that and what you learned. Then you're going to watch the endocytosis and exocytosis video talking about bulk transport with active transports and write down notes on that. Then you're going to go back here, and again, if you've done this with me before, you can either copy down this website, or if you just Google search, these are both McGraw-Hill. You can, if you're McGrawHill.com, you can Google search McGraw-Hill and the name of this, and it should come up. And then on this one, you will also click on how diffusion works. And you're going to watch that video and take notes, that animation. And then you're also going to do facilitated diffusion, and you're going to take notes on that one as well. There's several little animations here that you want to take notes on to see how those different things actually work. And that should go at the end of your notes to turn in.